Hey there, guys. So today I'm obviously not here. So our goal is to just basically go over the cell organelles and kind of just make sure we have what we need to in our notes, practice a little bit, and then we'll be done for the day. You will have a quiz tomorrow. It will be open notebook over the cell organelles, over their like function and also their location. Okay, so make sure you know what they look like as well. So again, it's open note. So make sure you have everything you need to in your notes so that you can do well on the quiz. All right, so we last talked about prokaryotic cells and prokaryotic cells being bacteria. And bacteria are basically really simple organisms. They don't have a nucleus or membrane bound organelles. And so that's what makes them different from eukaryotic cells. Eukaryotic cells are much more complex. They have a cell membrane, they have cytoplasm, they have a nucleus, so a way that keeps their DNA like kind of nice and safe. And then they have organelles. So again, organelles are kind of like our tiny organs. So they function in specific ways. So our first organelle we'll talk about is the nucleus. Now, the nucleus basically is your control center of the cell because it contains all of the genetic information, and that genetic information is your DNA. Now, DNA can be found in two different forms. It can be found in chromatin when it's not dividing, and then when the cell goes to divide, that chromatin is going to scrunch up and turn into chromosomes. And so, again, the nucleus houses the DNA. Now, one organelle that you might not have on your chart that you want to add is going to be the nucleolus, and that's this part right here of the nucleus. Now, the nucleolus is responsible for making the ribosomes, and it's important for us to know that because the ribosomes then go to make proteins. Proteins are important because proteins then are enzymes are proteins. And we know that enzymes help decrease activation energy in our cells, and then they help like in every metabolic process that we actually go through. So on this slide, I would like you to go ahead and just pause and write down 1 through 6, and then just write a T or an F and try to answer to the best of your ability, and we'll go over the answers together. So press pause now. All right, let's look at our answers. So all cells have a nucleus. That is false. Prokaryotes are cells, but they do not contain a nucleus. All cells have a cell membrane. That's true. All cells need a way to protect themselves from their outside environment or keep everything encapsulated in their environment. Question 3, eukaryotic cells are the only cells that have flagellum. Flagellum are basically like the tails you see on the end of sperm or the tails you see on the end of bacteria. So that one is a false answer as well. And we'll talk a little bit more about those later. The nucleus contains the cell's DNA. That is true. Chromatin will become chromosomes. That is true as well. So chromatin will condense and be organized before division happens. So mitosis or meiosis. The nucleolus makes the cell's DNA. Hopefully you have that as false because the nucleolus makes ribosomes. So our next organelle we'll talk about is the mitochondria. Mitochondria is another one to pretty much remember because it's the powerhouse of the cell. So what it does is basically take sugar, an energy source of the cell, and it transforms it into a usable form of energy, and we call that ATP. We'll get to the specifics about ATP in our next chapter. Just know that it's a usable form of energy for our bodies. Now, a way to tell the difference between a mitochondria and some of the other organelles is basically it has these folds inside of it, and we call this the cristae. And these folds are really important because the more folds there are, the larger the surface area, and then the more reactions that can take place. So your mitochondria kind of looks like a little bean with a bunch of folds on the inside. Our next organelle is the Golgi apparatus, sometimes called the Golgi body. It just depends on the site that you look for it in. The Golgi apparatus is really important because it basically is going to process, package, and export materials, kind of like FedEx, or I think the group that did this one said the secretary, which I really liked that analogy. So basically, when the Golgi apparatus gets a protein, it's going to process it and package it, and then it's going to deliver it to whatever place it needs to be in the cell. And it delivers it in these little vesicles right here. So you can kind of see that they're pinching off here. And so we just call those vesicles. Our next one is lysosomes. And the lysosome is basically like the garbage bin. So it contains these digestive enzymes that'll help break down things that aren't necessary for the cell. And then also go through, or it's called the suicide sac as well, because it is the reason cells can go through apoptosis, which is a form of basically cell death, where the lysosome will lyse, where it will break open, and all those digestive enzymes will destroy everything else that's in the cell. And so for the greater good of the cell or the organism, sometimes cells will destroy themselves so that the rest of the organism can thrive. One sad thing about the lysosome is that if you don't have a functioning one, or you don't have some that are functioning early in life, so this typically we don't see it in older individuals because they don't live past 1 or 2, but we see a syndrome called Tay-Sachs, and it's basically this buildup of lipids in the brain, and then those lipids start to take over the brain, and it's a really sad disease, and we'll talk about it a little bit more later, but basically those waste products aren't being processed, and then they take over the brain, and then these poor little babies don't live until 1 or 2. They don't live past that age. And it's just, obviously, it's just really, really sad. But that's one reason why we need to think about and we need to know things like lysosomes and what they do so we can understand them and maybe help prevent Tay-Sachs. All right, our next one is going to be the endoplasmic reticulum. Sometimes we call it the ER. There are 2 forms. We have a smooth form, and that one's going to basically transport lipids. And then we have a rough form, and that one's going to transport proteins. Now, the ER, the way to tell the difference between the Golgi apparatus and the ER is the ER is found right outside the nucleus. So here would be the nucleus. And then the ER is right outside. So that's basically where DNA, a copy of it, is made into RNA. And then it's taken to ribosomes, either outside of the nucleus on the ER or somewhere else in the cell. And proteins are then processed and made. So this is where ribosomes will be, and a place where protein synthesis can occur. And then they basically transport to the Golgi apparatus so that they can be packaged and sorted later. Right, the next one is our cytoskeleton. It's really easy to remember because, well, it's the skeleton, so it helps maintain and support the shape of a cell. It helps with movement of the cell. Don't worry about knowing the difference between microtubules and microfilaments. Just realize that there are different types, but the one that we do have to know about is the centrioles, and that's these guys here. These are found in animal cells. And basically, they're used during cell division. They help separate, or they create the spindle fibers that reach out and grab chromosomes, and they pull chromosomes apart so that each new cell has its own copy of DNA, so its own genetic material. All right, so then our next one is the vacuole. Now, the vacuole is kind of like the water storage facility. So not only does it store water, it stores other like important substances. But if you're going to look for the vacuole in a plant, it's going to be really, really big and in the center of the plant. And what that does is it allows to put pressure on the outside of the plant cell membrane so that plants have to be sturdy. If they're not sturdy, think about it, a plant can't just jump out of the way. It needs to be sturdy so it can withstand the environment that it's in. So plants have really large central vacuoles, and animals have smaller little ones all around them. So on a test, I would never ask you which one's the vacuole on a picture unless I included a definition, because there's no way you're going to be able to tell this vacuole from a different vesicle or anything along those lines. So kind of just keep it in mind. But in a plant, I could definitely ask you about what is the structure here, because it's very distinguishable from the others. Okay, so one big thing that we have to understand, and a reason why we talk about these organelles, is because of the protein production process. The cell is like a factory, and it's a factory that produces proteins. So with that being said, proteins are important because they make enzymes. So our steps, and you do want to write this down. DNA, it has the instructions. That DNA is then copied and then sent to ribosomes, where ribosomes then take the DNA and they put it all together and they form this long polymer chain. Okay, and then the ribosomes build that, it folds, and then the proteins go to the Golgi where the ribosomes build